Okay, this is our presentation, MSH2 gene mutations and its effects on hereditary non-polyposis colorectal cancer, HNPCC. My name is Georgia Branch.
Lady Wadike.
And I am Tira Lacey.
So HNPCC, hereditary non-polyposis colorectal cancer, is a form of cancer that is the most common form of hereditary colorectal cancer, and it is inherited as an autosomal dominant syndrome, which is a result of a defective mismatch repair proteins. It accounts for 2 to 5% of all colorectal carcinomas, and 90% of all colorectal cancers in HNPCC patients demonstrate a high microsatellite instability. HNPCC is associated with other cancers such as GI cancer or reproductive system cancers, and increases in cancer risk are due to inherited mutations that degrade the self-repair capability of DNA, so the ability for the genes to repair themselves. Symptoms of HNPCC include blood in the stool, extended periods of constipation, changes in bowel habits, weight loss for unknown reasons, and diarrhea that is not a result of diet or illness.
Okay, so this slide is going to describe the molecular effect of HNPCC. As we said before, it's an inherited mutation of one of the DNA mismatch repair or MMR genes, and that seems to be a crucial factor. MMR genes usually make the proteins that identify and correct sequence mismatches that occur during DNA replication, but if there's a mutation in that, then the DNA won't be able to repair itself correctly. Basically, the mutation inactivates the MMR gene that leads to the accumulation of cell mutations, and that can increase the chance of malignant transformation and cancer. Research in this field have identified 7 distinct MMR genes, which include MLH1, PMS1, but more specifically MSH2 and MSH6. And we will be focusing on MSH2 in this lab. So MSH2 is a gene, and it's a member of the set of MMR genes. It basically provides instructions for making a protein, and it plays an essential role in the repair of DNA. It fixes mistakes that are made when the DNA is replicating in preparation for cell divisions. And it also it joins with 2 other proteins, MSH6 and MSH3. But they're both produced from different genes, and they form a protein complex. It basically identifies locations on the DNA where mistakes have been made during the DNA replication. And then another group of the proteins, the MLH1 PMS2 protein complex, repairs the errors. This slide is basically showing the location of MSH2. And then this is a table showing the different MMR genes and in relation to the frequency of HNPCC cases, and MSH2 specifically accounts for about 45 to 50%.
And now for the fun part, the MSH2 mismatch repair lab. Our experimental objective of this lab was to gain a deeper understanding of the effect that an MSH2 mutation has on tumor suppression, which may lead to the development of HNPCC in humans. Our hypothesis was the presence of MSH2 mutation will encourage cancer formation. The overall purpose is to identify which mutations affect MSH2's repair function. So part 1, we isolated the plasmid DNAs from 3 E. coli strands using the Zippy Plasmid Mini Prep kit from Zymo Research. Then 2, we measured the concentration and quality of our DNA samples using the NanoDrop instrument. And then part 2, we digested 3 DNA plasmids using restriction enzyme XBAL and ABAL, or also called XBAL and APAL.
start over.
No.
No one told you that. We said, okay, go practice your signs, and you say, you know what? Let's just do it.
This is why we need to practice. Wait, so that's a 1?
XBA1
Wait,
and
ABA1. That's definitely down. That should
It's a 1.
But you put it back to a 1.
It's a 1, like
You
a
put it-
capital.
you put it back to a 1.
It's like a Roman numeral 1.
Wait, did you stop recording?
Yes, I stopped recording.
Fuck.
Yeah, we gotta start over.
Okay, wait. Let me. Can I look over the? I'm looking over the slides.
I would have just kept going, been like, I'm
No,
sorry.